Okay, well, good morning, everyone. I'm going to talk today about some work that my group has been doing for the past few years, kind of trying to think about the evolution of reproductive modes and the evolution of sex from a relatively new kind of angle where sexual conflict plays a key role. So there's so many questions about the evolution of reproductive modes, but I think it's now pretty well established that both sexual and asexual reproduction have their pros and cons. So sexual reproduction, by which for purposes of this talk I mean reproduction with males, is associated with some costs of so reduced transmission of alleles and reduced lineage growth increase and various costs of mating. But on the other hand, it's thought to allow for more efficient selection against deleterious mutations, and so a lower mutation load, and perhaps enhanced evolvability and adaptability. Asexual reproduction, by which I mean, for purposes of this talk, reproduction without males, so including parthenogenesis, avoids costs of mating, and it allows for more efficient, higher rate of transmission of alleles, higher lineage growth, but on the other hand, it's associated with increased mutation load and perhaps reduced evolvability. But there's some really compelling theory, including some new theory that I heard at this meeting, suggesting that actually the best strategy is a flexible strategy of being able to switch between sexual and asexual reproductive modes so as to take advantage of the benefits of each one. And actually, if we look across the eukaryotes, my impression is that most eukaryotes can actually do something just like that. They can use various types of flexible strategies. But there are some really weird exceptions, some really weird outliers where sex really predominates. In fact, where flexible strategies are rare and obligate sex is very, very common. And this seems to be particularly the case in complex animals. And we really don't know why. That's what I'm referring to here as the paradox of obligate sex. This is even kind of weirder if you consider there's nothing about being a complex animal that makes it impossible to be flexible, because flexible strategies have in fact evolved quite effectively in some animal lineages. So for example, we have cyclical parthenogens like aphids or like Daphnia, where there's an environmental trigger that triggers a switch from one mode to another. And even more impressively, I think things like facultative parthenogens, where any individual female can deploy either reproductive mode. What's more, in many species that we would consider obligately sexual, if you read the newspaper, every once in a while you hear about parthenogenetic reproduction. So recently there was a report of parthenogenesis in, I think, alligators. Before that, there was a report in California condors. And actually, if you put all this together, there's a lot of evidence of rare parthenogenesis in things like lizards and snakes and sharks and many insects, many species of Drosophila. So it looks like there's variation for the ability to reproduce parthenogenetically in a lot of sexual populations. But it doesn't invade. It very rarely invades. And we don't know why that's the case. Why does this really seemingly optimal strategy so rarely invade? And efforts to answer this question traditionally, or not this question, but generally to understand the evolution of sex, have traditionally taken the approach of looking for hitherto overlooked benefits of sexual reproduction. And these approaches, I think, have done a brilliant job of explaining why sex exists. Why does sex occur? Sex is clearly advantageous in certain contexts, but I think we still lack a robust explanation for obligate sex. There's really, I'm not aware of any theoretical or empirical work that really convincingly demonstrates how obligate sex can outcompete facultative sex. But my group and some others have recently been considering a very different approach to this problem, where rather than assuming that obligate sex occurs because it's advantageous, what if obligate sex occurs because it's a kind of evolutionary trap? What I mean by that is that once males are present in populations, they're under very, very strong selection to achieve matings. And this has led to the evolution of all kinds of really nasty coercive mating strategies. So it's possible that if a facultative mutant arises in a population like this, it will still end up paying costs of resistance, costs of mating, and perhaps it'll be unable to reproduce asexually at all, and thus unable to take advantage of its abilities. So this is quite a difficult kind of a question to address empirically, because these are rare events. A rare mutant arises, what happens to it? Why doesn't it invade? Who knows? But a few years ago, my former student Nathan Burke and I argued that actually facultatively parthenogenetic animals are very convenient systems for investigating these kinds of questions. Because in these animals, any female, if she mates, will lay fertilized eggs that will hatch into mixed-sex broods, so both sons and daughters. But if she avoids mating, she will lay broods of unfertilized eggs that will hatch exclusively into females. So for those of you who know the horrible terminology, this is Theleotokous facultative parthenogenesis that I'm talking about. And so in these animals, if there's any conflict, sexual conflict over mating, the outcome of that conflict directly determines reproductive mode. And what's more, it's easy to manipulate reproductive mode simply by manipulating access to males and matings. And because these are quite counterintuitive systems for most people, can you see what I'm, yeah. These are systems in which males are like a separate subpopulation. So males can persist in populations if they're able to achieve matings at a high enough rate, but males could also just go extinct, leaving an all-female population. And that all-female population potentially could be reinvaded by males at some point or not. And a key question, if we want to understand the role of sexual conflict in these kind of systems, is how do males and sex affect female fitness? So is there a net cost of males and sex, or is there perhaps an advantage? Do males perhaps, if males reinvade an all-female population, could this perhaps result in something akin to a genetic rescue? Because these all-female populations might have really low genetic diversity and so forth. So for the past few years, my group has been working on these wonderful stick insects from the tropical rainforest of far north Queensland in Australia, in the area kind of shown by the red dot. And this is called the peppermint stick insect, Megacrania batesii, and these stick insects are facultatively parthenogenetic, so they reproduce in the way that I just described. And they live in a variety of rainforest habitats, like that shown in the bottom left, on host plants in the family Pandanaceae. And these insects, so when we first started working on them in the field, we were intrigued to discover a mosaic of mixed sex and all-female populations. And so the mixed sex populations here are shown in red, the red dots, and all-female populations are shown in green, and they're very, very distinctive. So when you walk into one of these mixed sex populations, you tend to see males right away. And many adult females have a male clinging to them. Males guard females for an extended period, sometimes for weeks at a time. Males are vicious. They engage in really nasty battles, often right on top of a female. They bite each other. They try to rip each other's legs and wings off. If you go to one of these areas marked by the green spots, you see only females, just sitting there peacefully on their host plants. And we've been returning to these populations every year for the past 6 years, and these sex ratios have remained stable almost everywhere. And I'll get to that in a second. So my student Soleil Miller, who's unfortunately speaking right now in a different room about the same system, has done a genomic analysis and shown that all female populations have arisen repeatedly in this system, independently, and that the formation of these all-female populations results in an essentially instantaneous dramatic loss of genetic diversity and heterozygosity, but the phenotypic effects are pretty slight and subtle. We're still trying to figure them out. And my student, Daniela Wilner, who will be speaking in a couple of hours, has been doing population crosses and looking at interactions between the sexes, trying to understand the role of sexual conflict in these systems. But what I'd like to talk about today is some totally new data, new analysis, still in progress, so please don't photograph it and tweet it or whatever. Because focusing on a male invasion that we've been observing over the past few years in this central part of the range. So this is the only place where we've seen a change in sex ratio over the past 6 years. And this is interesting, firstly, because it's, I guess, kind of unusual to see an invasion in progress in a natural population. But secondly, because we can use that male invasion to look at the consequences of the introduction of sex into an all-female population. Does this result in something resembling a genetic rescue or not? So this is kind of what things looked like. So when we first came to this location, so I've zoomed in now on this location, the central location. You can't see my pointer, I guess. In 2019, we saw this. So along the coast, and the coast here, we've divided into several sections, separated by small estuaries. We found all-female populations along the beach in these areas N and S. Just north of that, in the area BK, there was a mixed-sex population, and just inland, just a few hundreds of meters away, there was a mixed-sex population. We came back the following year, 2020, same thing, no change. We came back the following year. Can Can you see that? Yes, I think. Yeah, so there was no change, but we realized something interesting was going on because eggs that we collected from one female in this area NN hatched into a mixed-sex brood. So we realized either that female had invaded in from a mixed-sex population or a male had come in from a mixed-sex population and mated with that female. And when we came back the following year, we found adult males for the first time in this area NN. We were very excited and shocked. And the following year, 2023, now males had spread through that entire area, NN. And there were even some males in area NS, but the sex ratio there was still very female biased. And this year, during the breeding season, NN had been completely invaded. Sex ratio is now essentially even. But area NS was still perhaps in the process of being invaded. Sex ratio was still very female biased. So what about the phenotypes? Well, we've been phenotyping these animals every year in these natural populations by photographing them in situ on their host plants. We don't collect adults. We try to avoid collecting adults. We photograph them without touching them, and then we extract phenotypic data from the images. And so a couple of relevant phenotypes that we can look at to try to test to try to understand the consequences of the male invasion are deformities, which we often see in wings in these animals. So these are flightless. Both sexes are flightless, but the normal wings are on the far left. They're held together. These are female wings. And then you can see slight and severe deformities shown. And we also see a lot of ectoparasites and infections. And the ectoparasites involve biting midges, like you see in one of the images there. We see mites. We also see a lot of infections that look like black spots on different parts of their bodies. And when these get really severe, they can just literally eat away at a limb or an antenna. So we can look at the rates of these deformities and infections and ask, has there been any reduction as a result of the male invasion? So if there has been something like a genetic rescue, we might see a pattern like this, where comparing this established mixed-sex population BK and this newly invaded population NN, we would see a pattern of decline in NN over the course of the male invasion. So the area prior to the invasion I've shaded in green, and the area in the last couple of years, in advanced stages of invasion, is shaded in red. So NN would these problems would decline to the same rate as in BK. So here are 4 phenotypes that I've plotted. And as I hope you can see, there is no evidence here of any decline in any of these problems. And putting all these things together into a single index, again, it doesn't look like there's been any sort of improvement. Life has not improved for these females. They still have just as many deformities, just as many parasites following the male invasion, now that the population has been invaded. And so to conclude, in this population, there's been no reduction in deformities or parasites or infections as a result of the male invasion. And this is consistent with the idea that males and sexual reproduction might actually be imposing a net cost on females. So I'd like to thank my students, my collaborators, the Australian Research Council, my institution, and the organizers of the meeting.
So, the sexes often experience different selective pressures. And one example, for instance, in Drosophila melanogaster, is that males and females have different optimal wing shapes. However, the sexes also share a genome, and this can create problems when at a shared locus, males and females this may create a problem when at a shared locus, alleles have opposing effects between males and females. And we call this intralocus sexual conflict. For instance, you can imagine if we have this one wing locus and 2 different alleles, one optimal female wing shape allele and one optimal male wing shape allele, well, this can lead to situations then where neither of those alleles can actually fix, because any increase in the frequency of the female optimal allele can be detrimental to fitness when expressed in males. And this can actually constrain the sexes from achieving their preferred genotype. However, it's not actually known to what extent the sexes experience this level of constraint, how they're experiencing it, or where they're experiencing it. And what I was really interested in exploring was what kinds of genes are the most constrained by the shared genome. So if the problem of interlocus sexual conflict is this shared gene pool, similar to some previous studies, what we wanted to do was then try to create separate gene pools. And how we did this was in the lab. We have these populations where we took a pool of genetically variable second chromosomes, and we labeled them with a DS red fluorescent marker, and I'll call these red chromosomes. In each generation, we selected males so that they expressed one of these red chromosomes, and females to be non-red chromosome homozygotes. So each generation, all the genotypes could be produced, but of course, due to the selection regime, the pool of red chromosomes are only passed father to son, experiencing male-limited selection and segregating like a Y. If the problem of intralocus sexual conflict, then, is this one genotype has to satisfy these two different selective pressures, while the pool of red chromosomes doesn't have to do that. They can just worry about male fitness and male selective pressures, increasing male beneficial alleles, even if those alleles are detrimental to female fitness. On the other hand, the pool of non-red chromosomes, spending 2/3 of its time in females, experiences female bias selection. We would expect that pool to increase the frequency of female beneficial alleles, even if they may be male detrimental. So we had 6 replicate populations in the lab and 130 generations of selection. We can look at genes where allele frequency has diverged between these two pools as evidence of genes where there's selection for this divergence, but they're constrained usually from actually acting on that. So these diverged genes can represent genes that are previously or usually constrained by the shared genome. If we do some Go enrichment analysis, we find that our diverged genes are indeed enriched for traits previously known to be affected by intralocus sexual conflict, such as development, locomotor activity, and wing morphology, as I mentioned before. So one of the first lenses that I wanted to ask this question, what kinds of genes are the most constrained by the shared genome, was to look at sex-biased gene expression. Sex-biased gene expression is just differences in expression between males and females. So unbiased genes are not dimorphic. Then you have male-biased genes, express more in males, and female-biased genes. And if the problem of intralocus sexual conflict is this one genotype has to satisfy these two different selective pressures, it's relatively straightforward to imagine that at a given gene, males and females might have different optimal expression levels for that gene. But if they can just develop sex bias and produce different levels of expression for that gene, doesn't that mean that intralocus sexual conflict is just resolved? Not necessarily, since just because there are differences in gene expression doesn't mean that either sex is actually at their optimal expression level. In this vein, Cheng and Kirkpatrick might hypothesize previously that, well, unbiased genes may be unbiased because of mostly experiencing sexually concordant selection, and perhaps at extreme levels of sex-biased gene expression, expression is sufficiently diverged that intralocus sexual conflict may be resolved or at least not constrained, but you might still expect to see a lot of constraint of sex-biased genes at intermediate levels of expression. So this was something that I wanted to investigate of whether or not sex-biased gene expression actually indicated a gene was more likely to be constrained, or if it was simply just a product of now intralocus sexual conflict has been resolved. So for each of our diverged genes, we can look at the level of sex-biased gene expression that it has and take a density distribution of those genes, essentially being able to compare relative amounts of male-biased genes to female-biased genes. And I can also do that in reference specifically to an appropriate null distribution of sex-biased gene expression. So now the y-axis would represent density relative to a null, where positive would mean that there would be enrichment compared to a null, and negative would mean that there's an underrepresentation. And if I do that, then the first thing we can see is that at these extreme levels of sex bias, we don't see very much difference in prevalence between extremely biased genes in our diverged gene candidates compared to the null distribution, suggesting that extremely biased genes are not particularly likely to be constrained by intralocus sexual conflict. However, if we look at more moderately biased genes, we see that male and female bias doesn't behave the same way. There's an enrichment of moderately male biased genes, suggesting that they are very likely to be constrained by interlocus sexual conflict, but we actually see an underrepresentation of moderately female biased genes, suggesting that their expression, it's not just that they're not particularly likely to be constrained, they're specifically unlikely to be constrained, meaning they're unlikely to have diverged between these candidates potentially, and that means that their expression levels or something about them may be more conserved in expression than the null distribution would suggest. So overall, does sex-biased gene expression indicate a gene is more likely to be constrained? Well, in most cases, no, but moderately male-biased genes are still experiencing a lot of constraint from interlocus sexual conflict. The next lens through which I wanted to ask this question of what kinds of genes might be constrained by the shared genome is to look at something from quantitative genetics and to examine the intersexual genetic correlation, so RMF, for gene expression. So in general, quantitative genetics predicts that a high intersexual correlation would mean that there would be more difficulty for the sexes to evolve independently. If increased sex bias is what may resolve interlocus sexual conflict, well, then we can imagine that for a gene with low RMF for expression, that gene has no problem developing sex bias and being able to have males and females diverge in expression until they each reach their respective optima, thereby resolving the conflict. But a gene with high RMF may have difficulty in becoming sex biased and therefore may be unable to resolve interlocus sexual conflict when it occurs. So we might expect then that our diverged genes would have an elevated RMF compared to a null distribution. So right now I'm just showing the mean RMF of the null distribution, and if we add the mean RMF of our diverged genes on top of that, we see indeed it's elevated compared to what you might expect by chance. So that's great, but I did show 2 slides ago, diverged genes are also enriched for moderately male biased genes, and moderately male biased genes are known to have a higher mean RMF compared to other types of genes. So I wanted to see and make sure that these were sort of separate things, and the elevated RMF was not just reflective of differences in sex-biased gene distribution. So I made this same comparison over 7 different sex-biased gene categories. So starting sort of in the middle, we'd be looking at unbiased genes. Then as we move more outside, we have moderately male biased genes, strongly male biased genes, extremely male biased genes on one side, and then the same moderately female biased genes, et cetera, on the other side. And if we're only looking at the null, which is plotted here, we can see that in the null distribution, as expected, you have higher mean RMF for expression for these moderately male biased genes. But if we add our means of mean RMF for diverged genes in each of these categories, we actually see that diverged genes have elevated RMF in 2 main categories, which is unbiased genes and moderately female biased. These categories are not overrepresented in our data set, so this suggests that the elevated RMF is not just because of this differences in sex-biased gene distribution, but occurs on top of that. And further, it may point to an indication of at sort of lower levels of sex bias, so unbiased genes or moderate bias in the case of female biased genes, that there are potentially some genes that are prevented potentially from resolving interlocus sexual conflict by becoming more sex biased. So overall then, what kinds of genes may be constrained by interlocus sexual conflict? Well, high intersexual genetic correlations for expression may constrain divergence and prevent the resolution of interlocus sexual conflict when it occurs, especially with low levels of sex bias such as unbiased genes. Moderately male biased genes are also very constrained, it seems, but extremely biased genes are unlikely to be constrained, and female biased genes may be more conserved in expression. In addition to looking at sort of sex-biased gene expression and RMF, I examined a few other different categories of what kinds of genes may be constrained, including functional categories and conservation among other species. But in the sake of time, and assuming that I was going to talk a lot slower than I ended up talking, I cut those, though I'm happy to talk about them later. And so I just wanted to thank everyone in my lab, and take any questions if there are some.
if that's our chime or the next room, but I guess I'll get started anyways. So thank you guys for coming to my talk. I'm really excited to share some of the cool work that we've been doing over the past couple years. Oh no, how do I switch slides? So despite a largely shared genome, males and females often look remarkably different, and this is really cool to me. One of the most common forms of this sexual dimorphism is sexual size dimorphism. I have pictured here one of the most extreme examples that I know of, the blanket octopus. In this species, females are 40,000 times bigger than males, and it wasn't until 2002 that we even knew that these two animals were the same species. Blanket octopuses seem like they would be a nightmare to keep alive in the lab, so I work with Drosophila melanogaster. Instead of 40,000 times bigger, the female's about 15% larger than the males. And previous work has shown that if you allow the male genome to evolve independently of the female, and then reintroduce that genome into females, not only do you see a reduction in female relative fitness, suggesting possible conflict, but you also see a reduction in that sexual size dimorphism with smaller female body sizes. So if we just generally hypothesize that the naturally occurring sexual size dimorphism in Drosophila melanogaster is due to a resolution of body size optimum and the conflict that incurs, then we could make the prediction that if we force this species out of the naturally occurring sexual size dimorphism, we may be able to reintroduce or introduce new conflict into the genome. And if we can do that, maybe we can actually observe what that conflict looks like, phenotypically and in the genome. And we have a really cool system to do this in. So back when I was just 13 years old, nervous about going to high school next year, Andrew Stewart started a long-term selection lineage that he's still maintaining today, where he took this geological sorting machine, and instead of geology, he sorted Drosophila with it by pumping CO2 in and sieving for body size. He has 4 separate selection regimes, 2 replicates each. Of course, he has a control where he sorts all the flies, but then doesn't select for body size. We have a selection on large size for both sexes, small size for both sexes, and then what I'm particularly interested in is a line where females have been selected to be small and males have been selected to be large, a reversal of the naturally occurring sexual dimorphism. Where the concordant selection lineages happened large and small, they responded really rapidly, within tens of generations. But the discordantly selected lineages took over 100 generations to reverse, and even more before they stably reversed their sexual size dimorphism. The work that I'll be talking about today happened when these lines were at almost 400 generations of selection, and I believe that they're almost at 500 now. So it's a really, really cool system. So the paper that Andrew and Bill Rice, Stewart and Rice 2018, initially published was showing general size on how they sorted through the sieve. So first we wanted to actually measure traits and see if we're getting trait-specific sexual dimorphism reversal, and if the other lineages, the up and down, were also changing sexual dimorphism just because they're under such strong selection. So here I have contrasts of sexual size dimorphism between the 2 concordant lineages and the discordantly selected lineage. And you can see despite the large and small being under such strong selection, they don't change in sexual dimorphism in the thorax, femur, tibia, or tarsus. But the discordantly selected lineage relative to controls reverses sexual size dimorphism in thorax, femur, and tibia. So despite we're just sorting them through holes in a sieve, we're getting a response from a bunch of different body parts, which is really neat. So I'd like to take this time to show some completely unrelated results from a completely unrelated experiment, just to give you an idea of what I expected in the first year of my PhD when we would look at genetic differentiation. I started and I was looking at these plots from another PhD student in our lab, and I figured, okay, we're gonna sequence these discordant lineages and we're gonna get a beautiful peak, a little bumpy landscape, one nice peak that tells us exactly where the locus under selection is. I was told by the PhD student and my advisor, this is probably polygenic. So I figure, okay, 2 or 3 peaks. So this is the first comparison that I did between discordant and control lineages, and we definitely got 2 or 3 peaks. It seems that the nature of selection also had a fair bit of drift, but I think we can still safely say that this is a very polygenic response. With how we have currently done it, we can't sort that drift and the genes that are responsible quite yet, but I'm going to talk about some future work where we're actually going to try to tease that apart that I'm really excited for. So we also wanted to look for this conflict by comparing male and female genetic differentiation between the sexes. And so I'm showing here some simulations that we did. We did 100 simulations using SLIM to control for the way that we were sampling, the history of these lines coming from the wild, and then being lab adapted for many generations, and then going through the type of selection that Andrew is putting them in every generation. I'm showing here one chromosome. On the top is the highest FST that we got when we compared between males and females within the simulated chromosomes, and then 2 sort of more typical FST distributions. The top graph, the dark purple line, is the maximum value that we got, and the lighter purple line just under it is the 95th quantile from all 100 simulations. And then the red line that's sort of hard to see that really trails 0 is just a trend line to give an idea to make it easier when we have all these dots on the plot where they're really laying. And when we look at our controls, male versus females, this is control replicate 1. We see a little bit of bumpiness, but really that red trend line it's just hugging 0 the whole way across. There's not any real big trends. There's a few windows that might be showing something interesting, but the trend is 0. But one of our discordantly selected lineages, just one replicate, shows a really clear peak in FST between males and females, which is really interesting because they shouldn't have FST that's not 0 if we were to catch them in the wild, because they're constantly sharing their genome with one another. The extreme selection that they're going under, and the fact that every generation there are 2,000 or more flies that are just brought down to the largest and smallest, 160 males and females, is really introducing a cool selection regime, and it appears really interesting conflict in the genome. So we also wanted to see if there were any other possible signs of this conflict that we can observe. So we decided to take all of both the concordant and the discordant lineages and cross them back both reciprocally to the ancestral population that the experiment started with, LHM. So I have highlighted here the discordant replicate 1 and discordant replicate 2. They're denoted with E for experimental. It's because of the original paper, I kept it consistent. And when they're crossed to the LHM ancestral population, their sex ratio generally is 50-50, like we would expect. But when they're crossed to each other in the F1 generation, the sex ratio starts to deviate away from 50-50. And this is for both replicates, not just the one that we saw that really cool male versus female FST change. Oh, and when they're crossed back to the ancestral, we don't see a change in the sex ratio. And in the F2, both the F2 generation of the discordant replicate 1, where we see the change in FST, crossed to itself in the F2 generation, we still see a deviation in the sex ratio, but also the F2 from one male sires of that discordant selection were mated to LHM dams, had a deviation in the F2 generation away from a 50-50 sex ratio as well. So this could be further evidence that we are seeing conflict in the genome and observing sort of the outcomes of that. So, of course, we looked for genes, and I feel like how a lot of these experiments go, we got thousands and thousands of genes because we're looking at really wide windows and really polygenic effects. We sorted with multiple things, so I've shown FST today here, but we also did CMH tests, we did some modeling, and we looked at nucleotide diversity, and we looked for things that really overlapped, that kind of popped out when we looked at anything. And then further from that list of hundreds of genes, we sorted on flybase for genes with a known sex-specific body size effect, and a sample of some interesting ones that came out, DMIC or diminutive, insulin receptor, so some things that are really, that we're excited for, that look like they have really cool potential. For the male versus female comparisons, previous work done in the ancestral population, LHM, has been done by Ruzicka and colleagues in 2020, looking for genes under conflict using a different method, but we had one gene that overlapped, Foreman-like, with Ruzicka et al. As a conflict gene. It has no known body size effects that we could find and no known sex-specific effects, but it's interesting that it's overlapping despite the fact that these LHM is diverged from our lineages by like hundreds of generations. So I showed you that really messy FST plot that gave me a shock in the first year of my PhD. But immediately after that, shortly after that, we decided that to really try to break apart this drift and allow recombination, we've set some crosses, reciprocal crosses between the discordantly selected flies and our control flies, as well as the large and the small. We've let them recombine for 18 generations, and we've actually just recently got sequencing back for multiple size classes that we phenotyped after 18 generations to see if we can look for alleles that are segregating differently based on size, and excitingly, maybe ones that are segregating differently in the two sexes based on size. So I'm really excited to get those results out, and maybe I'll be talking about it next year. And with that, I'd like to acknowledge everybody, and I think I have time for questions, which is exciting.
Well, welcome to my talk. I'm Susu from Michigan State University. So today I want to talk about the evolution of sexual dimorphism in Drosophila species. So if we think about sexual dimorphic traits, what can pop up in our mind? Examples here for you. Lion's manes, peacock's tails, and the flamboyant body color of the fish, and also fly's body size. Yes. So what I want to point out here is these sexual dimorphic traits, easy thinking about is they're not directly associated to reproduction because they're not reproductive organs. And they have been categorized as secondary sexual dimorphic traits and have been suggesting to become possible mating signals. So in these examples, lines, mane, darkness do affect the female mate choice. Also, the number of eye spots in peacock has a correlation with the peahens they mated with. So again, when we are thinking back about these sexual dimorphic traits, what are we missing here, or what novel insights we can provide as like a current stage of research? So the first thing popping in my mind is these researchers have been focusing on morphological traits who display sexual dimorphism. No offense for body size, but the physiological traits we have been care about less. And the second is, again, if you think about this, sexual dimorphic traits usually can be seen as male executive traits, and all of those examples like peacock tails, lion's manes. About female executive traits, we have been less studied as well. So these are the questions I'm trying to address and provide insights in my studies. So the system I'm using is Drosophila, Drosophila cuticular hydrocarbons, CHC. So during the courtship ritual, a male will try to encounter a female, and if, like by tapping with the foreleg, they could perceive a series of chemical signals they have been recognized as CHCs. So these CHCs perceived through the foreleg tarsi can act as contact pheromones in Drosophila species. A well-studied case for the mating signals as CHC in Drosophila is Melo-Gavin. Melo-Gavin and its sibling species, Drosophila simulans, have a very strong pre-mating reproductive isolation barrier. And this is simply because of the production of a female and the Drosophila Melo-Gavin specific CHC, the 711 heptacosadine. So this CHC can stimulate, as working as aphrodisiac, to stimulate the courtship ritual from the male Drosophila Melo-Gavin, and it will inhibit the courtship ritual from the heterospecific Drosophila simulans males. So the first question with the system to be asked is, again, can we use the system to understand how the degrees of sexual dimorphism in a physiological trait with the CHC? And also, I further want to ask, can we possibly use the degrees of sexual dimorphism to predict the potential role of mating signals in Drosophila species as well? A quick catch-up for the term degrees of sexual dimorphism was have been used like by evolutionary biologists for a long time, and back in 1994, study on New World monkey species, and they already started using the term of sexual dimorphism, degrees of sexual dimorphism, by comparing the body weight, again, morphological traits, where you could see like they have a different degrees of sexual dimorphism. So the first part I did is we selected 9 species across the Drosophila species, and we used the GCMS to analyze the CHC profiles, where you could see here, they definitely show a great variation for CHC profile sexual dimorphism across the phylogeny. From the left to right, less different to more different, and Drosophila erecta has the highest degrees of sexual dimorphism. And on the contrast, Drosophila pseudo-obscura has the least degree of sexual dimorphism. So the next step I did is to test whether these Drosophila species do use CHC for, as a mating signals for recognition or no. Because Drosophila males use the phorolactacy to perceive the CHC information, so I did a micro surgery on them to remove the phorolactacy and perform these no-choice mating assay to see the courtship initiation, if it's got affected or no. So this is like courtship reinitiation in Drosophila. One more time. Yeah, basically it's like a vibration the wings and play some songs to attract the females. So the result, quick result here is in Drosophila melanogaster, after removing the male phorolactacy, the courtship initiation was not being affected. Drosophila simulans, sibling species, after removing the phorolactacy, got affected. Among all 9 species, where you could see Melanogaster, and there's down like the second last one, Drosophila repleta, after removing the phorolactacy, no significant decrease. And the other 7 species across the phylogeny, they do show a significant change in courtship initiation. So tarsi remove do affect the perception of CHC in males and also affect the male initiating their courtship ritual. But we're suspecting, possibly because of the tarsi removal is not solid assay enough, maybe because they just affect the movement ability of the males. So we use another assay to validate our result. In the lab, we can generate a transgenic line of fly without producing any single CHC compound. We call them naked fly, and we can extract the CHC from other species coated on these naked fly, and again to test the courtship initiation to see if CHC is the key to initiate the male courtship attempts or initiation. So we selected the species with the highest degree of sexual dimorphism, Drosophila erecta, and the naked fly with Drosophila erecta CHC can significantly stimulate the erecta male's courtship initiation, and the same as the least degree of sexual dimorphism, Drosophila pseudo-obscura. So this finding validated our previous findings about CHC play a key role in the species, in some Drosophila species, for initiating the courtship ritual. So, and the very last part for this part of the study is we did a correlation analysis to compare between the degrees of sexual dimorphism and the changes in courtship ritual to see if there is any pattern. And however, negative data, there is no enough evidence to support there. We could use the degrees of sexual dimorphism to predict the role as mating signals. Okay, so now it's concluding the first part, so I'm moving to the second part. So the second part, I'm more interested in this species, Drosophila erecta, displays the highest degrees of sexual dimorphism in CHC. Why this is happening? Why erecta is so different? We always care about the difference, right? So when I go back to check the phylogeny where Drosophila erecta is in, which is Melanogaster subgroup, by comparing the major CHC compound they produce, all of the males in Drosophila melanogaster subgroup produce the same major CHC in males. In females, well, we could expect some variations, right? This is my storytelling is gonna go. So simulants, Yakuba and Arena, are producing exactly same CHC as their males. Melanogaster, Seychella, a little bit longer CHC, and the Erecta, even longer. Okay, this probably is not enough yet, but Drosophila erecta is very different already. And then we did a PCA analysis where you could see most of the male CHC profiles and a couple of the female CHC profiles all clustered together. And the most distinct and distant cluster is the Drosophila erecta female CHC profiles. One more information to validate you. If we compare the Drosophila erecta and Drosophila arena CHC profiles, GCMS tells us, or tell you, they are very different. This is why erecta shows this high level of the degrees of sexual dimorphism in CHC. Each peak stands for single CHC compound, and where you could see that the erecta female is representing a very strong or exaggerated sexual dimorphic trait. So my second part of the question is, what is the genetic mechanism underlying here in erecta CHC production? And also, this is so much fun because it's a female exaggerated trait model for us to understand. What we know about CHC synthesis is they are synthesis in inocytes. Inocytes, like in this diagram, the gray part, the gray area are inocytes. And also, they share fatty acid synthesis pathways. Again, we care about the differences in chain lengths of CHC. Drosophila erecta is producing. So I am specifically curious about a specific family of enzymes involved in this pathway, elongase genes. And we hypothesize that at least one elongase gene is involved in the production of these very long CHC. So to identify the candidate genes, literature search is always being helpful. Melanogaster has been well studied. In Melanogaster, for producing a long CHC is the elongase gene called ELO-F. And is it present in Drosophila erecta? Is the ortholog of ELO-F responsible for this? So we went back to the erecta genome to see, to understand about the ELO-F ortholog. And the result of the synteny comparison is the ELO-F is present in some of the species, but it's not present in erecta or arena. So now we're getting into like a, dead end. We have nowhere to go. So the next way we have to do is we got the genome information for all 19 elongase genes in Drosophila erecta genome and use ISICA in situ hybridization to test the expression in inocyte. And where you could see the purple dye, here are a few examples of the purple dye indicates the expression of the elongase genes. You could see some of the genes are expressed in both sexes. Some of the genes has a male bias expression, and among all 19 elongase genes, we have only one gene, the last one, 517, has a female bias gene expression, and here is a better picture for you. So the 517 is the candidate gene. So we further asked the question whether this female bias expression is due to cis regulatory change in Drosophila erecta. So in the genome, we found this candidate gene, and also we found the regulatory region. The assay we are doing here is we molecular cloning this piece of regulatory region coupled with the GFP reporter gene, make a construct, use the GFP as an assay to report the function of the regulatory region we're testing. So firstly, the ORI-A, what I'm showing here is the regulatory region of the ortholog of this candidate gene in arena, where you could see is very weak GFP expression. And the erecta-A, the erecta regulatory region, can drive a weak in male GFP expression. And of course, it's a strong expression in female. So cis regulatory region changes do contribute to the female bias expression for this candidate gene. And also, the next step, we separated and narrowed down the regulatory region of EREA into A1, A2, A3, test the GFP expression by building the constructs one more time. A1 and A3 drives no GFP expression. A2 can drive a monomorphic expression. Weird. So why this is happening? We had a further hypothesis. It's because there's an individual sex-related module, which could be in A1 or A3, so we simply did 2 more constructs, A1 plus A2, A2 plus A3, GFE expression. A1 plus A2 shows the monomorphic expression, and A2 plus A3 can shows a female bias expression. So then we have the conclusion that the sex-related module is in A3. So a quick recap or summary for my study here today is, firstly, CAC profiles across Drosophila has a great diversity, variation in degrees of sexual dimorphism, but currently I don't have enough evidence to tell you that degrees of sexual dimorphism in CHC can be used to predict the CAC roles as mating signals in Drosophila species. And also, Erecta shows the highest CAC sexual dimorphism and as a great model as female executive traits. And we found a candidate gene, and it's due to cis regulatory changes. Thanks for all the help.